So what I'm going to start out with are some of the technologies that we're on the cutting edge right now. And this is what is called an example of pre-implantation genetic diagnosis. In this particular case, this is a very happy family. This little boy is named Noah. He is the child of these two parents in Britain. What happened is that the mother has a genetic default, a disease called neurofibromatosis, and she would pass it on to her kids. So she decided that she did not want to do that. So she went to a fertility clinic in the UK and had some extra eggs produced, and they were fertilized with her husband's sperm, and then they had 13 different embryos. Each one of the embryos was tested to see whether or not they had the gene for this particular disease. It turned out for her, 12 of the 13 embryos had the gene. If they had been implanted, they would have the disease. So they took the one that did not have the gene, and fortunately, they have a child, Noah, born without that disease, without that defect, who will be healthy. And all of his children down through the ages will never have to suffer from that disease. This is what it's now capable of doing. I've just checked, this is a process that is also used in Russia. Several thousand babies a year are born using this technique as well. So, all right. Where is the machine? Okay, we'll keep hoping. In this particular case, this is another technology that's just been approved in the UK, in the United Kingdom, and this is supposedly what they're calling it, the three-parent baby. This is not something, it's not really three parents. What is actually occurring is that some women, unfortunately, have defects in what are called the mitochondria. These are the little organelles in each one of our cells that produce energy for our cells. And we inherit them from our mothers. They do not come from our fathers. And one of the problems is that how would you fix this defect? Well, what you can do is take the nucleus, most of the DNA, most of what makes us, our bodies what they are, and put it into the donor egg, where the mitochondria are healthy. Then you have the reconstituted egg, and you have that fertilized with the sperm of her husband, and a child is born, presumably without these particular defects or these diseases. There's something like 1,000 different mitochondrial diseases out there, so this is actually a problem that exists in the world. This has just been approved in Britain. It actually occurred in the United States in the year 2000. There was a hospital, St. Barnabas, which did this particular technique and basically was able to have 20 healthy children born using the technique, but our regulators decided that it was too scary or something and decided to ban it. Fortunately, it looks as though this technique will now be used to help parents, again, have healthy children using this kind of technology. Now, a more interesting thing that's coming down the pike, researchers have now been able to do this. This was done in China in 2016, is basically to create sperm using skin cells. Basically take one of your cells from your body, treat it in a certain way so that it turns itself into what are called induced pluripotent stem cells, and then you treat it further and they become sperm cells. And they've done this for mice. In fact, they've done it for mice and created mice eggs as well. And so they've been able to create healthy mice using skin cells as transformed into sperm. Now, this technology is going to be is on its way to human beings as well. All right, there we go. This actually is in April of this year. This was just reported in Scientific Reports, which is a major publication. Spanish researchers have made that achievement for human beings as well. They've taken skin cells from men. One of them was 78 years old, by the way, and they were able to transform those skin cells into sperm. And they know that it works. They have not obviously tried to use it at this point. The safety considerations are very important, but it is a proof of concept. And the reason I'm coming up to this is you'll see that this is a technology, all right. I'm being defeated by technology. Help. Nope. Nope. Biology's supposedly harder than this, but anyway. There we go. No, that's backwards. Forward. No, backwards, forward. Try this way. Oh. Thank you. All right, we go back now. All right, so this is going to be important, and this is a technology we have to take into consideration. This is called genomic sequencing. The first human genome was sequenced for the cost of $500 million in the year 2000 in the United States. There were two teams that did it, a private team and a federal government team, and they raced to the finish line and agreed to a tie. It cost $500 million to do that. Right now in the United States, you can have your genome sequenced entirely for $1,000. It's fallen that fast. All 300 billion of your stems of your genes can be base pairs can be sequenced and it can be known. The costs, according to some of the people I'll be talking about later, will be down to $10 by the end of the decade. $10. You would be able to spit into a tube, give it to a laboratory, and in a couple of days they would come back and say, here is your genome, and you would have that information available to use as you would desire. Now, the idea is that these technologies, using skin cells to make germ cells, eggs, sperm, and so forth, plus this genomic screening, is something that Henry Greeley, who's a bioethicist at Stanford University, is predicting will occur in the United States and other rich countries in the next 20 to 40 years. He thinks that actually half of all babies will be conceived in 40 years in the rich countries using this technology. It will work the following way. A couple will decide they want to have babies. They will go to a clinic. They will have their skin cells manipulated, turned into eggs and sperm. They will create, using these manipulated eggs and sperm, 1,000 different embryos, or 100 different embryos, let's say. They will have them all tested using PGD, the genomic screening test that costs now $10 a test. And then the doctors will go to them and say, well, you have 100 different embryos. Some of them are more athletic than others. Some of them have a better academic possibility. Some of them have a musical ability. Some of them, whatever possibilities, a range of possibilities that these embryos will have. How tall they might be, whether they might suffer from early onset Alzheimer's, so forth, will be available to the parents to decide. Now, this is not genetic modification. This is genetic selection. Basically, parents will be given a wider range of choices about the sorts of children they decide to have. And Greeley is fairly convinced that this will be a very cheap process by the year 2040 to the year 2050, and that most people in rich countries will choose to do this because that way they can be more sure that the children that they are bearing will be healthy. Because you will also be checking to see for defective genes as well as you do this process. So he calls this easy PGD. PGD. And I don't know if he's right about this, but it does seem like a plausible possibility. But of course, something like this technology would be very useful for people who are already infertile. About 15% of couples around the world have problems with fertility. So if you are able to use your skin cells again to make sperms and eggs, that would certainly help those people who are infertile to have children that are genetically related to them. Other possibilities are that you might be able to do this for same-sex couples. For example, two women might be able to go in, have their skin cells turned into a sperm and an egg, and put together into an embryo so that they too would be two mothers of the same child. They would share a parental relationship, a genetic relationship with their child. You could do that as well for obviously male couples. But then those are still more or less genetic selection. Those are not genetic modification. You're not picking and choosing what genes people might have. In the last 3-4 years, one of the most amazing technologies ever, something that we often use the word disruptive, but this is an incredibly disruptive technology called genome editing has come into existence, and it's come into existence from the research of two researchers, a French researcher, Emmanuel Charpentier, and Jennifer Doudna from California, University of San Francisco, California, where they have discovered how to basically edit genomes with great precision and very easily done. In fact, I've ordered a kit that should be on its way to my house where I will be able to use this technology to manipulate and edit the genomes of some bacteria on my kitchen table. I hope nothing bad happens. I don't think so, but I'll get back to you on that. So this is something that's coming down the pike. Now, this is the case of a young girl named Layla Richards, also in the United Kingdom. In her first year of life, she came down with a very bad form of leukemia. There are all kinds of conventional treatments that people tried to use to help her, to cure her, and they were failing. So her parents went to the physicians and said, is there anything you can use? And so in this particular case, they had a technology where human T cells, they are part of the immune system that behaves badly in the case of lymphoma, just happened to be on the shelf. Well, you can't put T cells from one person into another person and have them work. The immune system will not allow that to occur. So what they did was use a genome editing technique called TALENs to change the genetics of these T cells so that they matched her immune system. And so they replaced her immune system with these manipulated and genetically modified T cells, and it worked. She is apparently cured of lymphoma. And this is a case where these technologies will be used to cure people who are already existent, who are already suffering from genetic diseases. You might be able to use these technologies to fix them after they're already alive, as they did in the case of Layla here. The technologies we're talking about, there are a whole bunch of different ones out there, and they'll be all very complementary, but the key ones that people are talking about is called CRISPR. It was derived from essentially the way bacteria protect themselves from invasions of viruses. It turns out that bacteria also get diseases. And what happens is that this is a technique for basically for a bacteria to, when it's invaded by a virus, to chop it up so that it cannot infect it. And so it's a very precise way of identifying foreign genes, and now has been adapted for editing human genomes, genomes of plants, animals, everywhere. It is adopted all over the world in thousands of labs. It's possibly the most widely adopted new biotechnology in the world. There's a newer version of this that just came out in the last year or so, also derived from bacteria. It's even more efficient, even more precise. I think it's called the Skol-Koval Institute of Science and Technology have been working on this as well, and with the folks at the National Institutes of Health in the United States, and they have optimized this as well. And it is also coming into technology. So one way that this technology might work, one of the things that happens as you get older is that your organs wear out, or you have heart disease, or your liver fails, or you have diabetes, and so forth. One prospect for using this technology, and it's already been a proof of concept, is that you could take a pig embryo, manipulate its genes using this technology, edit its genes so that they match human genes, so that your immune system would not recognize an organ transplanted from a pig as being from a pig. It would be accepted into your body and not rejected. This is one of the problems with transplants. If you have an organ donor, someone whose organ you're using, you always have to be sure that it matches immunologically, and you have to take a lot of drugs to make sure that it doesn't get rejected. This idea is that you would be able to grow organs that would match human immune systems perfectly and not need the drugs. Now, again, we're not at that stage, but George Church, who we'll be talking about later, who's at Harvard University, reported as a proof of concept again in November that he had edited all at once 62 genes in pig embryos, and that had worked. So this is a way to do this, that you'll be able to not just do one gene at a time, but whole genomes edited at a time. Another way that pigs might be useful, also using this technology, and this is work that's being done at the University of California, Davis, is essentially what you do is you take, again, a pig embryo, 8 cells, whatever, 16 cells, and you make sure that it can't create a particular organ. Say it can't create a heart, or it can't create a pancreas. And then what you can do is a human being who needs a heart, you could take a skin cell from that person, you could turn it into what are called induced pluripotent stem cells. That is a stem cell that can become any tissue. And then you take those cells and you inject those cells into the pig embryo at a very early stage. And what happens is they segregate themselves to make the organ that the pig itself cannot make. Because you have disabled the ability for the pig embryo to make a heart or a liver. Now these cells will be differentially segregated to become a human liver or a human heart growing in a pig. And then you would be able to take, as you know, when the pig is a certain size, the good news is pigs are about as big as we are, so their organs kind of match, is you would take that human heart that now matches the donor, the recipient, and you would be able to use that as a transplant. Again, using the CRISPR technology to genomically edit the pig's genome so that it now matches or enables the possibility of matching a human genome. Now, we get to the designer babies, the problem that a lot of people are worried about, which I will try to explain later why that isn't a problem. Back in 2015, a Chinese lab was capable or succeeded in basically editing the genomes of some non-viable embryos. They were not viable because they were triploid, they had too many genes, but what they were trying to do was to say, can we use this CRISPR technology to fix a gene for a disease called thalassemia? And it turned out they could. It wasn't very efficient. They did manage to get out of perhaps 100 or so embryos. They only got it to work perfectly in 3 of them, but it did work. And they were able to correct the disease gene in those particular embryos. Then another team in China, just again in April, announced that they had tried doing this to confer HIV resistance to the virus that causes AIDS, in also non-viable embryos, and it was much more efficient this time, and so they're moving down that path. This is Professor Nayakan. She is at the Francis Crick Institute in Great Britain. And what she has applied for and had been granted permission to do is to try these techniques, CRISPR techniques, genome editing techniques, on very early stage human embryos. She's not going to try to implant these embryos or anything. She's basically trying to figure out questions of why embryos don't implant, how the growth goes wrong, and so forth. It's a medical thing. She's trying to figure out how to help people solve fertility problems. So again, this is very early stage, but this is the kind of thing that will be developed over time. It's one baby step at a time. So, do we get to this someday? Well, we'll see. One of the things that will first be happening, if we're going to use genome editing at all in human beings, in embryos and so forth, it will be used to fix genetic problems first. You might start doing it for example, if a couple knows that they have a genetic problem, they might decide to have actually their gametes, their eggs and their sperm, edited using CRISPR, and then combine them into an embryo that would no longer have the defect that they were trying to avoid. So what will happen first if we use CRISPR, and I think that we pretty much will be using CRISPR to do this, will be to fix problems. Not enhance people, but to fix problems, to get rid of diseases, to get rid of defects. And we know that's the kind of thing we know to do because we can identify the defects. We know how to fix broken things far, far better than we know how to improve things that are not broken. So the first steps will be figuring out how to fix diseases. And I think that will most likely occur in the next 10 years or so, if not sooner. But then this is the prospect down the line. Do you want to be able to pick traits of your children? Would you not just pick the traits, but would you like to add traits? And again, the problem is there's a lot of information about the genome that we don't know. And so it's very hard to say, well, actually, if you put in this gene, so-and-so will become a Nobel Prize-winning physicist. It doesn't work that way. You might be able to figure out how to make someone slightly more athletic. You might be able to make someone more musical. You might be able to make sure that they have a much longer life than average, those kinds of things. But it's going to be much harder to figure out how to make someone a super genius, supermodel soccer player. That's just going to be very hard to do. But it won't necessarily be impossible. But what it will do is give parents options to figure out ways of helping their children to flourish and ways they think are valuable for their children to lead good and fulfilling lives. Interestingly, just as I was coming over here today, I was reading an article in Molecular Psychology where, just today, it just came out today, where some researchers in the United Kingdom have done what are called genotype screening tests. They tested 5,000 people for 10 million differences in their DNA, all 5,000 of them, and they were able to correlate 74 differences in genes that are responsible for as much as 10% of the variance in people's academic success. It's kind of amazing. Again, that's a lot of genes, and not everybody knows what they do at this point. These are just correlational studies, but this is already on the way to figuring out how our genomes actually operate. This is an interesting particular thing. For example, if you are deciding to use these technologies, perhaps you'd like to know what your future child will look like, right? This is Craig Venter. He is the guy who raced the government and nearly won the race to first sequencing the human genome in the United States. And his particular company is called, and we'll be talking about it a little bit later, is Human Longevity, Inc. What they can do, he claims, is to take your DNA, sequence it, then put it through a system of algorithms that he has, and then predict what your face looks like. In this particular case, this is the actual face of this particular person, and this is what the algorithm shows. All right, it's certainly not perfect at this point, but his claim is that he would be able to show parents, and it's going to get better over time, parents what their kids might look like at age 18. So you could take an 8-cell embryo and say, in 18 years, this is what Bill is going to look like. I don't know. But there are some interesting forensic consequences of this as well. We all shed DNA wherever we go. This includes criminals. It might be possible to find this DNA and sequence it and then get a police sketch of a person and know what they look like as well. And it could be useful in that way for apprehending criminals. Then there is this interesting project called, we sequenced the Human Genome Project, that would be called Human Genome Project READ. We're reading the data. This new project was announced back in June, last month, is called Human Genome Project Write. The idea is that you would be able to take chemicals off the shelf and create a complete human genome from scratch. All 3 billion base pairs, and not necessarily because you're trying to create a synthetic human. This is not necessarily the goal. So far, it's only been done in bacteria and yeast, and so very simple single-cell organisms. But the important thing about this is that if this is done in the next 10 years, which is the goal, it will provide researchers and physicians tools for figuring out all kinds of ways of intervening in people's genetic health, both those of us who are still alive and also for people, children and embryos in the future. So this is going to create the toolkit that will make the possibility of designer children more probable over time. Of course, there are people who object to these things. And now we'll discuss some of these people and why they are wrong, or why I think they're wrong in any case. This is Richard Hayes. He is with a sort of left-wing think tank in California called the Center for Genetics and Society. And his particular concern is that what will happen is the rich will get these technologies first, They will create themselves into a genetic elite and leave the rest of us all behind, and we will become serfs to a genetic elite. I don't think so. First of all, the technologies are all going to take a while. It's not going to happen immediately. It's not going to be a whole suite of technologies that come out at one time, and you get to apply them, and all of a sudden your kids are Superman. This is not the way this kind of thing will work. There will be step after step after step, and some of them will be failures. But the other thing I think will be happening is that as these technologies roll out, they will become cheaper over time. They will become more available to everybody over time. What I think will be happening is that parents who want to use the technologies to improve the lives of their kids, to give their kids, if you will, good characteristics that other children get naturally, parents will be able to confer that on their kids as well. I think that, in fact, ultimately what happens is that it reduces inequality rather than enhances inequality, because these technologies will become fairly widely available over the course of this century. There will be no genetic supercaste running the world. This is Michael Sandel at Harvard University, and he has this notion that if we interfere with the lives of embryos and children to be, that somehow we are not reverencing the gift of life. I think about it this way. Nature very handily deals out to millions of people every year disease, disability, early death. Now, what is more reverential of life than trying to prevent those outcomes for people, trying to prevent those outcomes for children, to give them the possibility of avoiding debilitating diseases or early death, to give them the possibility of flourishing over time. I think it's far more reverential to use human intelligence and technologies to improve human lives rather than say you just have to be stuck with the genetic lottery that you got. That is not reverence for life. That is fatalism. This is Marcy Darnowski. She's also at the Genetics and Society Center. And her particular concern is, well, medical concerns and also social concerns. The medical concerns are real. You certainly don't want to deploy this technology until we're pretty sure that it's safe to use, that you're going to get the results that you want. And I think a pretty good benchmark for that, and it's the one that is basically used in in vitro fertilization techniques now, is it more dangerous than natural conception? About 2% of all children born on planet Earth have some sort of genetic or other flaw, and if you can get the use of this technology down to a risk of about that level, I think then you can call it safe. And it's not there certainly by this point, but I think that gives a benchmark for doing it. So her medical concerns, all right, there's a way to deal with that. I think it will be dealt with. People are not wanting to use this technology if they think it will harm their children. They want to use it if they think it will help their children to be. On the other hand, her social concerns are, again, the inequality issue, and I don't think that it's a real issue. Again, another way to think about it is, sure, some of the rich will be using these technologies first, but one way to think about it is that they're lab rats for the rest of us. They're testing out improved technologies that will work if they don't work, well, sorry. They'll be able to pay for the consequences. This is a German philosopher, Jürgen Habermas, and he has a particular concern which I frankly don't understand at all. His concern is the following, is that if your parents pick your genes, that somehow means that you are not the author of your own life. This is a deep puzzle for me, why he would think this. First of all, I will ask anyone in this audience, did anyone in this audience consent to be born? Okay. Did any of you consent to be born with the genes that you happen to be bearing right now? Okay. So my argument will be as follows, that essentially children whose parents use these technologies will, to give them certain qualities and genes and so forth, will stand in exactly the same ethical relationship that you stand in relation to your parents. Now, the other thing that's implied by Habermas, which I find very odd, is that somehow genetic ignorance is a kind of freedom. The fact that you don't know what your genes are and where they came from and how you got them randomly makes you freer than if you do know where they came from. That's completely backwards. Actually, I don't get it at all. The more information you have, the freer you are, because you can overcome the kinds of things that particularly are conferred upon you. Your mind is not at the mercy of your genes are at the mercy of your minds. I'll give you a couple of examples from my own life. As it turns out, I've had a genotype screening test. I've not had my genome sequenced. That's coming in the next year. But a genotype screening test looked at about 1 million different single nucleotide polymorphisms, basically differences in my genes and other people's genes. And I posted all that information online. So you can go to Snipedia, put my name in. It's called Snipedia. Put my name in, and you can find out what's wrong with me, if you're interested. But I discovered when I went through that I had a much higher propensity to be addicted to cigarettes than most people do. And for a fact, in my 20s and 30s, I was a 4-pack-a-day smoker. I am no longer a smoker. I haven't been smoking for over 25 years. Genes are not fatalism. They are not determinism. Knowing information, I think, frees you rather than imprisons you. And so this notion of randomness as freedom is completely backwards. And even if it is, the fact of the matter is that everyone in this room in the next 10 years or so will have all your genes sequenced anyway, and then what happens to your freedom? Habermas is quite clearly wrong. On the other hand, I have to say that many of my fellow Americans do not agree with me. This is some polling data from Harvard University, where, much to my horror, 2/3 of my fellow Americans decided that they wouldn't even want to use these technologies to help children overcome genetic diseases. It's 2/3. Worse yet than when they were asked, well, what about improvements? What if they could be used to enhance people? 80% said, no, we do not want to do that. All right, so perhaps I should despair. The history is not on my side. That's completely wrong. Let me give you an example. In the 1970s, if you had taken the similar polls and asked people about in vitro fertilization, test tube babies as they were called in those days, the majority of people would have said, no, absolutely not. This is against God's will. We can't possibly do this. And I could go back and cite all of those polls. In fact, I did cite all those polls in my book. And what happened? All this opposition, a lab in the United Kingdom finally was able to succeed with in vitro fertilization. And Louise Joy Brown was born in 1978, I believe. And she was perfectly healthy. Her parents could not have been happier. They had been trying for years to have a child. And within 2 years, the public had completely shifted the other way. They became very much in favor of this. In vitro fertilization clinics were proliferating all over the place. Success is what will change people's minds. If the technology works, they will change their minds, and they'll change it very, very fast as well. So I'm not worried about these polls at this particular point. So let's go back to consent for a moment. This is by a philosopher named Fritz Allhoff, who's written some really, I think, very thoughtful analyses of these issues. And one of the things that he points out is, what is it that parents are likely to want to do for their kids using these technologies? Do they want to make their kids stupider? Do they want to make their kids less healthy? Do they want to make them weaker? Do they want to make them less musical? Do they want to make them uglier? No. What parents will want to use these technologies for is the exact opposite, to give them agility, to give them, to the extent possible, more agile brains as well, to provide for better health, to overcome the possibility of terrible diseases, to enhance the possibility of them living long, healthy lives. So his notion, and I think he's completely correct in this regard, is that we can presume the consent of people who are going to feel themselves healthier, smarter, perhaps more beautiful. Consent can be assumed because everybody wants to be smarter, prettier, more athletically agile and healthier. This is what these technologies will do. What about those who oppose them? What moral burden do they bear? What should we think of them? Well, this is a philosopher at Oxford University, Julian Savulescu and his colleagues. And this was written in response to people who were objecting to that Chinese study, the first one in 2015, where they had tried to modify embryos for the thalassemia gene. And I think his point is quite salient, very strong. It's basically, if you prevent these technologies, you have the burden on your head of condemning millions of people to disease, debility, and early death. If you oppose these technologies, you are responsible for harming people who could have otherwise lived better, fuller lives. And I can't think that it's a stronger point could be made than that. This is George Church. He is something of a phenomenon in the United States. He's at Harvard University. He's got his fingers in every bit of biotechnology. He's most recently working with a company called Editas, which is at the forefront of the CRISPR technology now. And he said, I think this rather amazing thing that we're going to be able to reverse aging in 5 to 6 years. I hope he's right. Given my age, I do hope he's right. I would love to be 30 again. But I think he may be a little optimistic on that point. But I think he's right that eventually the technologies will be able to do that. There are companies already working on that, again, in the United States. There are several, but Google is spending several tens of millions, maybe hundreds of millions of dollars in a company called Calico, California Life Company. And then Ventnor, which we met earlier, is Human Longevity, Inc., is also working on these technologies. Right now, there are a couple of interesting products. This is from a company called Elysium. The one thing that we do know that increases mammalian lifespan and mammalian health span, and I hate to say this, is calorie restriction, eating less food. If you feed animals, mammals, about 80% of what they would really like to eat, they live longer, they have less heart disease, they're more cognitively available. This is for rats, this is for monkeys, it's every species we've tried it in. It works. I actually have some friends who are doing calorie restriction. You have never seen as skinny people in your life. And someone has joked is calorie restriction may not increase your lifespan, but it may feel like it. But so what if we could do this in a pill? And this is what Elysium is trying to do. They've discovered, they think, the various pathways, the metabolic pathways that could be activated where you could trick your body into thinking that you were on calorie restriction. And they are offering a supplement that they claim will do that. I'm not taking the supplement, but I'm thinking about it. There's another company called Alkahest, which is working on, when I'm in a certain kind of mood, I call it the vampire cure for aging. As you know, vampires, by sucking people's blood, live forever. Well, as it turns out, it works. This is what is called parabiosis. And what you do, and what has been done, and it works very well, is you take an old mouse and a young mouse, and you stitch their circulatory systems together. And it turns out that by doing so, you make the old mouse younger. The heart rates improve, their cognitive abilities improve, their muscle mass improves, all kinds of things are improving. If they have a glucose problem, that also improves. And so something in young animal blood can rejuvenate people. So what the company Alkahest is doing right now is an experiment. We don't know the result of it. They're taking several Alzheimer's patients and are transfusing blood from young people into them with the idea of checking to see if this has any effect on their disease and if it has any effect on their aging processes. And again, this has just started, so no one knows. It's a proof of concept thing. My best guess is it won't work, but the other possibility is that would it be possible to isolate the factors that are in young blood that create these that do the rejuvenation. And one of the compounds is called GDF11, and some people are working with that now. The problem with GDF11 is that it's very controversial. There are some people who think it does nothing. In fact, they think it might do harm, and others who are very, very big on it. But this is science. We'll find out. And if it's not GDF11, there's something else in young blood that might be isolated, that might be useful. So we'll see. But then again, there are people who don't want you to live long, radically healthy lives. This is Francis Fukuyama. He wrote a book called Our Posthuman Future. He and I have debated several times. I always win. In any case, his argument, he was on, by the way, President Bush's Bioethics Council. His argument is that essentially it might be good for each one of us to want to live a long time, but it's bad for society. I'll deal with that in a moment. This is what I call a left-wing bioconservative. This is Daniel Callahan. He was the co-founder of the world's first bioethics think tank, the Hastings Center, in the late 1960s. He's now at Harvard University. And his statement is that you shouldn't be able to decide how long you want to live. Why should you get to decide that? So who does get to decide that? Well, according to Frank Fukuyama, your government gets to decide that. Essentially, his argument is that the government should take away from you the burden of deciding how long you want to live. That has never worked out in history very well, I would just suggest to you. Interestingly, one thing you have to think about is that without asking anyone's permission in this room, human life expectancy increased from 35 years in the year 1900 to over 70 years now. Basically, human life expectancy doubled, and nobody asked anyone's permission for that to occur. So why should anyone have to ask for permission if they want to double their life expectancy to 150 at this point? The three main arguments, there are others, but these are the three main arguments that people have against radical life extension. And those are, I basically refer to them as the nursing home world, which the idea is that somehow or other you'll be kept at age 80 for 150 years. The point of all this research is not to be older longer, it is to be younger longer. No one wants to be 80 in a nursing home for the next 150 years. So that's simply not what's going to be happening. This is not what the research is aiming for. The next one is the concerns about overpopulation, and I can understand why people would have that. But some calculations by a demographer, by a guy named Jay Olshansky, who's at the University of Illinois in Chicago, he made the following analysis that assume everybody on planet Earth was made immortal tomorrow, and we had the same fertility trends that we have now. What would the world population be around 2100? And he figured it to be about 10 to 11 billion people, 10 to 11 billion people, which is considerably up from the 7.4 that we are now, but it's not necessarily totally destroying the world. And I think the point is that it's not going to happen immediately tomorrow. We're not all going to be made immortal tomorrow. As these technologies come on, we will have the ability to make some sorts of decisions among ourselves, such as if you take these treatments, perhaps you don't get to reproduce. I don't know what the future will bring, but I have no doubt that humanity will be able to handle this problem. We don't need to go precautionary principle immediately on this particular issue. And the final one is, can we afford this? There was a fairly prominent article in a magazine in the United States called The Atlantic Monthly, which suggested, oh my goodness, what would happen if these longevity treatments cost $15,000 a year? It would wreck the entire healthcare system in the United States. Trust me, we're well on the way to wrecking it ourselves without this. Right now, we already spend about $15,000 a person in the United States on healthcare per average, and we don't get very much for it. Think about it this way. If you could get $15,000 to make yourself younger so you wouldn't have heart disease, you wouldn't have Alzheimer's, you wouldn't have diabetes, think of how much money that might save the healthcare system if everyone were young. The other thing to think about is this is going to be happening over the next century. Economic growth, unless our governments manage to screw that up entirely, is expected to be about 2 to 2.5% over the next century. And if that's the case, average per capita income on planet Earth will be from $10,000 per capita now to something over $100,000 per capita. And in rich countries, Western Europe, the United States, it's projected to be as much as $300,000 per capita. At $15,000 a year, that's what, 5% of your income? I think you might spend that on treatments that would keep you feeling 30 years old for another 100 years. I don't think unaffordability is a problem. So, what about you all? I notice there are a lot of young people in this room. And I'm going to tell you this, I tell my younger colleagues this, I think there's a very good chance that those of you who are under 30 will get to decide how long you want to live. These technologies will come on in time for you to take advantage of them. And this is something called the longevity escape velocity notion. As you get older, the technologies improve faster than you get older. So the things that keep going wrong get fixed faster and faster and faster. And a friend of mine who's a biogerontologist named Aubrey de Grey has famously said that the following, that the person who's going to live 150 years and a person going to live 1,000 years is only a 10-year difference between them. There's a very good chance, I would argue, that there's someone in this room who's going to be the first person to live to 1,000 years. In any case, this is my hope. I do hope that he's correct, that in fact, I'm not one generation too early. Thank you very much for this discussion. I look forward to your questions.